Hello, everyone, and welcome to today's webinar titled Spatial Transcriptomic Profiling of Spiny Projection Neurons in the Mouse Striatum. My name is Elizabeth Schneider, Senior Global Marketing Manager, and I'll be your moderator for today's event that is part of the 10th Annual Neuroscience Virtual Conference. Today's educational web seminar is presented by LabRoots and sponsored by NanoString. For more information about NanoString, please visit our website. Now let's get started. Before we begin, I would like to remind everyone that this event is interactive. We encourage you to first participate by communicating with other attendees using the new live chat feature during the presentation. You can find the live chat located at the right-hand side of your screen. You can also participate by submitting as many questions as you would like during the presentation. To do so, simply type them into the Ask a Question box and click Send. We'll answer as many questions as we have time for at the end of the presentation. If you have trouble seeing or hearing the presentation, click on the Help Desk button located at the bottom of your screen within the navigation bar or from the lobby of the event. Finally, as a reminder, this presentation is educational and offers free continuing education credits. Click on the Continuing Education Credits link located in the abstract window below the presentation window and follow the process to obtain your credits. I now present today's speaker, Alison VanSchoiack, Senior Product Enablement Scientist at NanoString Technologies. Thank you so much for being here, Alison. We're looking forward to your presentation.
Okay, thanks Elizabeth for the introduction. Today, I'll be talking about using spatial transcriptomics for detailed localized profiling of specific cell types in focal areas within the mouse brain by presenting data from both a project conducted internally at NanoString as part of our Spatial Organ Atlasing efforts, and another project described clearly in the title here that was looking into spiny projection neurons in the mouse striatum. That was done in collaboration with Dr. Caitlin Romano and Dr. Ellen Hess at the Emory University School of Medicine. The field of spatial biology is in many ways very new, as the genomics field has evolved from using bulk onto single cell to spatial methodologies within the last 15 years to understand the dynamic gene expressions at play in development and disease. It's clear that cell types, organization, and gene expression patterns play a critical role in how genes orchestrate development and disease, and really only spatial methodologies allow us to collectively ask the questions: what are the cell types, how are they organized, and what are the gene expression patterns at play? Or in this case, allow us to look fully at the fruit tart instead of its individual components or a blended product. Now, interestingly, I think the neuroscience community was really well ahead of the game in realizing the importance of spatial transcriptomics and also in execution. So the Allen Institute for Brain Science was founded in 2003, and one of their first priorities was mapping gene expression throughout the mouse and human brains. This was an amazing amount of work and a resource used by many I know in the neuroscience community still today, including myself, of course, along with their increasing number of other databases and resources. These are incredible resources, the scale, throughput, and expense, so the whole institute dedicated to these atlasing efforts, not really practical for most investigators to use the same methodologies to conduct genomics experiments in the same way in the context of disease or using a disease model of interest. It's certainly far from the originally intended scope of the atlasing efforts. Now, excitingly, almost 15 years after this first spatial transcriptomics experiment was published, spatial transcriptomics was named as Nature Methods Method of the Year in 2020, which in many ways means that today's spatial transcriptomics methods are available to any researcher to use to better understand spatial gene expression in the context of disease or in a given disease model. So this is what spatial transcriptomics transcriptomic profiling looked like in 2006. So essentially the Atlas released in 2006 has in situ hybridization images for over 20,000 genes throughout the mouse brain at 200 micron intervals. So we can see in the bottom right side of the slide an example of the gene DRD2, and this is just a subset of the different coronal planes that were profiled in this effort. So we can clearly see the expression pattern of this single transcript throughout the striatum as get deeper into the brain. Now, for any one of these given coronal planes that we're looking at, there are roughly 20,000 in situ hybridization images on that same exact plane, showing transcript by transcript spatial resolution or spatial expression with high resolution in each of these images. Now, importantly, this is spatial, but it's not necessarily cell type specific. So we see DRD2 expression in or transcripts in the striatum, but we can't necessarily associate them with a given cell type from this resource. Still, an amazing tool, amazing reference to use. Now, 15 years later, we've come a long way in making these spatial methods accessible for targeted whole transcriptome level experiments. So the experiments that I'm going to discuss today use the GeoMx Digital Spatial Profiler, which enables users to visualize their tissue, select regions, features, or cell types within regions of interest for whole transcriptome profiling using, in this case, our mouse whole transcriptome, but we also have a human whole transcriptome assay. Now, step by step, how this works is that you would first stain your slides with fluorescently labeled antibodies, or you could use in situ hybridization probes as well, along with our GeoMx oligoconjugated RNA detection probes. These are hybridization probes that are conjugated to a barcode, or that have a barcode conjugated that includes a UV photocleavable linker. So essentially, once the reagents are on the slide, the instrument will take an image of that slide. You can select your regions of interest on the tissue, and then the instrument will use UV light to cleave the barcodes in any region of interest designated on the given sample. So at the end of the experiments, these are then collected into a 96-well plate, and then at the end of the experiment, for whole transcriptome assays, the barcodes are read out on an Illumina sequencer. Now, the magic behind the system really lies in the incorporation of the digital micromirror device that's sort of shown here in this cartoon type of image. There are actually two of these in the GeoMx instrument, each containing about 1 million teeny tiny mirrors that enable the system to shape the UV light to directly match the biology in a given tissue section. So in this example, we have an image, a region of interest from a mouse brain, selected from mouse brain, stained with 3 different neuronal subpopulation markers along with a microglia marker. And the instrument can make a mask based on the fluorescent signal from each of these different markers. And then the DMD can shape the UV light directly to that mask to specifically illuminate each population separately and collect a whole transcriptome worth of information from each of the different populations. So this is just one way of targeted transcriptomic profiling in the brain. Here we've listed multiple other approaches that can be taken. So you can use geometric profiling, which enables the fine selection of different histological features within a given tissue. Here we show the selection of different cortical layers in a mouse brain. You can use segmentation, which can enable where contiguous features can be selected or separated using fluorescence. In this case, we're looking at granule cells versus the Purkinje layer in the cerebellum. Rare cell or cell type specific segmentation, as I showed on the previous slide. A common application in the neuroscience community is to use a contouring type of approach where a specific feature is identified on the tissue section, in this case a beta amyloid plaque identified, and then can contour out from that feature on the tissue at a specific distance to profile the microenvironment around the plaque, sort of in step-by-step increments. We can also do gridded profiling or even combine gridding with with another approach outlined here. So we could profile the pink neurons within this image here on the far right, for example, independently of the remaining tissue in each box shown on the grid. Now, with so much flexibility, it can sometimes make it challenging to design a good experiment, especially as these tools for generating high-plex spatial transcriptomic data are becoming more mainstream and relatively easy to use. So one of the things that NanoString has recently put forward to help facilitate the spatial biology community is our Spatial Organ Atlas, which is a publicly available reference data repository for demonstrating new paradigms in spatial organ research. Thus far, the database consists of 5 human organs. We have lymph node, colon, pancreas, kidney, and human brain, along with the mouse brain data that I will be sharing today. And we have plans to expand to include additional organs for both human and mouse. One of the primary purposes of this resource is to establish best practices for spatial transcriptomics across different organs and tissue types. So I'll briefly walk through the mouse brain data set for the Spatial Organ Atlas today as an example of different ways to utilize spatial transcriptomic profiling, and importantly, to visualize and interpret the results. I'll follow this up with a great example from within the neuroscience community to show how spatial transcriptomics is being used for targeted research questions. So I'll start here with the Spatial Organ Atlas. On this slide, we can see an example brain image from the Mouse Spatial Organ Atlas experimental data set. So the reference data in the SOA, as we call it, is from 4 male 60-day-old C57 black 6J mice. We've profiled the same region from mouse to mouse. Most of the data I'll show here today is going to be from a single mouse. That's the way that we put the stories together on our website, but the data for all 4 mice is available for download at our website and can be explored from there. And for the GeoMx experiments, we visualized neuronal nuclei using NeuN, that's shown here in magenta. We visualized astrocytes using GFAP, shown in cyan, and microglia using IBA1 in yellow, along with a nuclear stain. The different regions with the shaded overlays in the image are the different regions of interest that were collected for this experiment. So in the bottom right cartoon, you can see all of the different areas of the brain that were profiled. And these areas were aligned on this coronal plane that we imaged, same plane for all 4 mice. We aligned the regions using a different atlas from the Allen Brain Institute, the anatomical reference atlas that we see in the top right. Now, as I mentioned, selecting good ROIs can be one of the most challenging aspects of targeted transcriptomics experiments. The brain is very complex. The mouse brain contains 100 million cells organized into at least 461 recognizable 3-dimensional structures, as annotated in the anatomical atlas from the Allen Brain Institute. Now, I stress recognizable here because we're learning that there are likely more regions than those that are visually distinct, and I think this will we'll discover more and more, especially as spatial biology becomes more mainstream. For the Spatial Organ Atlas experiments, we sampled from structures that are of common interest across multiple disease areas in the neuroscience community. One of the great things about GeoMx is that you don't need to select a single ROI strategy. For this experiment, we used both geometric and cell type specific segmentation approaches. So on this slide, I've highlighted only the geometric ROIs that were collected for this experiment. You can see here that we used polygons to trace distinct histological features like the cortical layers or the dentate gyrus of the hippocampus, shown here in this magnified image. Now, again, the GeoMx instrument can use that digital micromirror device we see in the cartoon to specifically illuminate these features based on their morphology. In many regions, such as the amygdala, hypothalamus, cataputamen, various thalamic nuclei, we've placed geometric circles to enable the collection of multiple consistent replicates within a given structure or substructure. These are 200 micron circles. It's technically still bulk profiling to some degree, but I really like this approach if you don't need to get super granular so that you can establish multiple replicates within a given area and can get really fine spatial resolution without the need for micro dissection. We also use cell type specific segmentation in the cortical layer ROIs and also in select regions from the cataputamen and the amygdala. In these ROIs, we use the NeuN signal to specifically segment and profile the neuronal nuclei in each of these regions. So in the image in on the right side of the screen, you can see a zoomed in view of one of the ROIs from the cataputamen. And you can see the magenta NeuN staining and then the orange overlay of the mask that was used to target the NeuN positive cells within the region of interest. We also collected what we call here the neuropill region from each of these ROIs that was segmented. In this case, the neuropill contains the synaptically dense area of tissue between the neurons. And here we also, this segment also does include some glial cell bodies and processes. I wanted to highlight here that we also were able to collect from the choroid plexus, and we did so using sort of a combined segmentation geometric type of approach where instead of trying to match the fine tracing of this very unique, or to trace this fine unique tissue structure, we used a geometric shape to designate a region of interest in each of the ventricles, and then use tissue autofluorescence to select only the tissue in that region. Okay, so some data here to share. The plot on the left shows the total number of genes detected throughout the different regions profiled. Each color is a different primary region, such as the thalamus or hypothalamus, and then the different shading, the colors are shaded to designate all of the different subregions. In those ROIs that were segmented into NeuN positive and neuropill segments, we see the NeuN positive segments in yellow and the neuropill segments in this sort of reddish orange color that you can see across multiple of the different regions. Now, as might be expected, we did see more genes detected in the neuron-rich segments across all of the regions profiled, with about 10,000 genes on average detected in each of those neuron populations. On the right side of this slide, we're looking at a heat map of subset of general gene sets that would be expected to be expressed in the brain, specifically in the regions. We're looking at these gene sets and regions collected that have been segmented into NeuN positive versus neuropill. So a few things to point out here. First is that in the NeuN positive segments, we see an enrichment of gene sets involved in like chromatin modification, mRNA splicing, gene sets that are involved in transcription translation processes, which matches what we see on the total number of genes detected. Neurons are more transcriptionally active than the other cell types or tissues that are tissue areas that are profiled in this experiment. Because we did include some glial cells were included in the neural pill components, we see an enrichment of glial-related genes in the neural pill segments. And then synaptic genes, as may be expected, we see an enrichment in cortex specifically in both the NeuN positive and neuropill segments. This may make sense given that we know many of these genes are locally translated at synapses, so we would expect to see them in both the NeuN positive and neuropill components. Interestingly, sort of in the middle of this plot, we see serotonergic synapse genes are enriched specifically in the neuropill, which may be indicative of more locally translated components at this specific synapse type. So another thing we can look at with GeoMx spatial transcriptomic data is cell type deconvolution of the spatial data. So here we have an overview of non-cortical ROIs. The cortical ROI data is available on the SOA website, just didn't fit directly here on this slide. But the way that the cell decon works is by aligning the GeoMx data with pre-existing single-cell RNA-seq data to identify the cell type composition of each ROI collected. So these stacked bar plots show the proportion of each of the different cell types referenced, in this case, in the DropViz mouse brain single-cell RNA-seq atlas. From this plot, the differences in choroid plexus versus the parenchyma, I think, are quite striking, as we would expect, with very high alignment of choroid plexus and endothelial tip cells in the choroid, and more neurons and glial cells in the parenchyma. Now, within the parenchyma, the white matter ROIs collected from the corticospinal tract. These are a little bit hard to see on the image, but they are the circles in gray that are overlaying the cyan GFAP positive area between the thalamus, hypothalamus on the right side of the image. So these white matter ROIs collected from the corticospinal tract are very different from those that were placed in gray matter. As we might expect, we see high numbers of oligodendrocytes in these regions, as well as other glial cells. If we look at the NeuN positive segmented ROIs, we can see a clear enrichment of neurons in each of these populations, while the neuropill segments contain more mixed population, more glial cells in those populations. Still a decent neuronal component. Again, we expect some local translation of neuronal genes in the neuropill segments as well. And then the remaining ROIs here are all geometric, and as we expect, we see a mix of neuron and glial cell types. So here we're looking specifically at cortex, and we can see the different cortical ROIs selected on the right side of the slide in the image. The principal components analysis on the left side shows a nice separation of the different segmented cell populations, with the geometric segments in blue in the middle, clustering sort of in between the two different segmented populations, NeuN positive in yellow, and again, the neuropill in burnt orange. So given that the geometric segments are the sum of the two parts, it makes a lot of sense that we would expect to see them spot in the middle. The boxes on the right, these are layer 1 regions, and these are the cell composition of these regions is quite different from the other cortical layers and actually more reflects that of the neuropill versus anything else. Now, it's hard to see on this plot, but PCA2 is actually driving some degree of separation by cortical layer. I think this is actually nicer to look at with the whole cohort of 4 samples. So on this plot, the shape is associated with the population collected, and that pattern reflects exactly that which we see on the left side, the PCA on the left side from the single sample. And the different colors represent the different cortical layers. So you can see these different cortical layers sort of beautifully cascading down the y-axis, where we go from layer 6 in yellow to 5, 4, 3, 3, 2, and then 1 is kind of over there on its own because of its unique cell type composition. So continuing on in cortex, if we look into the NeuN positive ROIs, we can identify neuron-specific genes that are enriched in each cortical layer. The gene clusters, I think, are pretty clear on this heat map. This is a heat map from the whole cohort, so n equals 4 mice. And this is a heat map of the top genes that are differentially expressed between the cortical layers. So we look at some of the specific genes identified in this analysis. We can find spatial confirmation from that original spatial transcriptomics experiment described and published by the Allen in 2006. And it can be a lot of fun to use the Allen Brain Atlas to spot check GeoMx data, but I'll just pull out a few examples here of some of the key genes identified on a region by region basis. So here I'm showing CUX2. This is a homeodomain transcription factor that's well established to be enriched in layers 2 through 4. It serves as a marker of upper cortical layers, and we see that in our GeoMx data. The arrow shows where we see this gene within our GeoMx data set, and then on the left we see the expression pattern from the in situ hybridization for that gene in the Allen Atlas. Matches very nicely. Higher signal in the upper cortical layers. Next gene of interest is SAPB1. We see this most highly expressed in layer 4 in our data set, although there's some expression in layers 5 and 6 as well. And this is a gene that is an activity modulated transcription factor, which is enriched in interneurons, and it's established that layer 4 has a highest density of interneurons of any of the cortical layers, so it makes sense that we would see higher expression here. A little bit hard to see on the ISH image, but you can see the sort of granular layer clearly labeled, and then labeling in layer 5 and 6 as well. ETV1, we find expression exclusively in layer 5, and that's very clearly seen by this in this image. The in situ image, this is a transcription factor that's expressed in a subset of layer 5 pyramidal neurons. We see very clean expression in both the GeoMx and with the ISH. And then layer 6, we see this gene that's been reported as a marker of layer 6 neurons by others as well. And this is IgHM, it's immunoglobulin constant domain gene. No one's really sure what it's doing in layer 6 neurons, but it's present and can serve as a marker of those neuron types. So we see really nice enrichment of this gene in layer 6 in our GeoMx data set as well. So backing out of the cortex here, we can do the same type of differential expression analysis, and it can be very useful for identifying genes expressed by cell populations in other regions of the brain, which can allow us to begin to associate genes with functional behaviors. So in this example, we're comparing adjacent NeuN positive segments in the cauda-putamen versus the amygdala. So let me zoom in a little bit here. So we see the different, the neurons that are profiled in the cauda-putamen outlined here in purple, and the overlays are shown in orange, and then down in the bottom of this image, the yellow trace outlines the amygdala regions and the different neurons that were profiled, and those regions are shown in orange with the orange overlays here as well. So this is a comparison between two directly adjacent structures with very different connectivity and functionality within the brain. And we see that reflected with vast differences in gene expression profiles between those two different regions. I've highlighted some genes known to be expressed in each of respective population profiled here, such as the excitatory synaptic regulator, C1QL, that's shown enriched in the amygdala. And expression in the basolateral amygdala is known to be required for normal fear memories. And then on the other side of the plot, we see multiple genes that are associated with dopamine transmission in the striatum. Elevated, these are genes that are elevated here in the catoputamen, which would be expected for a striatal region. Again, here just showing some ISH verification. This is DRD2. This is one of the genes, the gene that I shared at the very beginning of this presentation. We can see nice expression in the catoputamen and none in the amygdala. We see that nicely reflected when we look at this gene specifically in that analysis. So again, DRD2, it would be expected to be in the striatums. So to some degree, low-hanging fruit here, but expect that there's a likely much more novel and interesting biology that remains in this data set to be explored. Here are some of these that are labeled and also some that are unlabeled. So I encourage anyone that's interested to take a look at this data set. It's all available for download at our website. And many of the plots that I shared here today are put together in a really nicely packaged step-by-step story. This is the opening page here. You can see the table of contents. This is accessible for anyone to walk through and really get a nice example of a GeoMx experiment from the mouse brain. Now, from here, I'd like to jump into the next experiment I'll be discussing, which this essentially serves as a targeted follow-up GeoMx experiment to the Spatial Organ Atlas. And here, we've worked with some collaborators at Emory who are very interested in looking at specific neuron populations and sort of atlasing these populations to the same degree from a distinct region of the brain. Essentially, we're looking at a slightly different, more rostral coronal plane from the same type of mouse brain. The specific neurons of interest in this project are the spiny projection neurons that sit within the striatum. So the spiny projection neurons are a special type of GABAergic inhibitory neuron that make up about 95% of neurons within the striatum. They're modulated by dopamine from the substantia nigra, and the classical difference between these two cell types is that they both control movement essentially the direct spiny projection neurons will promote movement via direct pathway, so directly projecting onto the globus pallidus and substantia nigra pars reticulata, while the indirect pathway spiny projection neurons, the ISPNs, work to inhibit movement via a more indirect pathway to the same target. So what's important here is that the DSPNs promote movement while the ISPNs inhibit movement. Now these two different neuron populations reside within the striatum. which is essentially this big light blue blob that we see on the reference from the anatomical brain atlas that's shown on the lower left side of the screen. Now, interestingly, unlike many of the other structures that we identified in the earlier experiment and that are shown on this same coronal plane, where we see subregions of within each structure, there's really no further stratification of the striatum on the anatomical atlas, at least thus far. Really no obvious cell patterning on the Nissl stain on the left side of that, the split image either. However, recent work has shown that there are actually substantial differences in connectivity and gene expression patterns when adding additional spatial context into the striatum. And in fact, it can be subdivided into at least 3 functional subregions. So this looks something like this, where we have the 3 different functional subregions. The dorsal medial striatum receives convergent input from associative areas, including the prelimbic area, posterior parietal association area, and the temporal association area. The dorsal medial striatum is thought to support goal-directed behavior by representing associations between responses and outcomes. The dorsal lateral striatum is thought to be involved in more habitual and skill performance, and it receives sensory motor input involving the trunk and lower limbs. The ventrolateral striatum also receives sensory motor input, but for the mouse at least, involving the upper limbs and mouth. Now, our collaborators at Emory, Dr. Caitlin Romano and Dr. Ellen Hess, they sort of ambitiously wanted to independently profile the 2 different neuron populations in each of these 3 striatal subregions that are shown here. Fortunately, the 2 different neuron populations can be readily identified using RNA-scope fluorescent in situ hybridization probes, as we can see here in this image. So for the GeoMx experiments, we use the DRD1 RNA-scope probe to identify the DSPNs, which are shown here in cyan. The gray image is Cyto83. This is a nuclear stain. The ISPNs are identified here using RNA-scope for ADORA2A, and this is shown in magenta. We can see from the merged image that these neurons are tightly packed in the striatum, but the signals don't overlap. So for each of the brains profiled for this experiment, there are a total of 6 wild type C57 black 6 brains profiled here, 3 male, 3 female. For each of these brains, we placed ROIs in each of the 3 spatial subregions of the striatum, as are shown in the top right image. So we have dorsal medial, dorsal lateral, and ventral lateral. So for a single mouse, we looked at 6 different cell populations total in this region of the brain that otherwise appears relatively homogeneous. So a quick recap here on the GeoMx workflow. As the same workflow as I shared previously, but in this case, the fluorescent RNA-scope morphology markers were used to highlight the features of interest in the tissue instead of fluorescently labeled antibodies. Other very minor differences were that multiple sections were placed and profiled on a single slide, as shown in the schematic, although I think Kate was able to get 6 total brain sections on a single slide. So this is a single slide. This experiment was conducted on a single slide, and a large data set from a single slide experiment. The other main difference is that the Spatial Organ Atlas samples that I described were all FFPE. These are fresh frozen samples, so the assay will run with either sample type. Otherwise, exactly the same as shown before. first plot I'm showing here is looking at gene expression changes in the DSPNs versus the ISPNs throughout the striatum. This is all of the subregions of the striatum collapsed together. Over 11,000 genes were detected in total in this experiment. Again, we're only looking at 2 different neuron types, so it's really nice number of genes overall that we're detecting. The genes that we see in each of these populations indicate clear enrichment for each of the population profiled. We see canonical ISPN and DSPN markers, those that we actually use for segmentation, DRD1 and Adora 2A showing up on the respective sides of the volcano plot, along with several other genes that are known to be expressed in each of the different cell types, providing a really nice sanity check that we're collecting from the regions intended, the cell types intended. Moving forward with the more spatial gene expression comparisons, we first compared gene expression patterns in the DSPNs specifically, noting 172 total significantly differentially expressed genes between the dorsal medial and dorsal lateral striatal regions. Between the dorsal lateral and dorsal ventral, ventral lateral regions, there were 356 differentially expressed genes identified. Looking at the ISPNs, we saw a total of 76 differentially expressed genes between the dorsal medial and dorsal lateral striata. And like with the DSPNs, we see a larger number of differences between the dorsolateral and ventrolateral ISPNs, with a large increase in the expression of ROBO3 in the ventrolateral ISPNs compared to the dorsolateral. So these data, we continue to we're continuing to work on further analysis for all of the work that's presented here today. But I think both studies really speak to the fact that the spatial transcriptomic profiling methods with DSP, as shown here, really allow investigators to ask targeted questions looking at both the region and cell type specific transcriptomics throughout the brain. Both of these projects today were collected in wild type or control mice, but both used approaches that could very easily be adapted or adopted by investigators that are working to investigate different mouse models of neurological disease, or even looking into different therapeutic trials using mouse as a model system. So all of this work was done in about 1 week total. We collected about 10 sections from sample prep to sequencing readout within that week. And I think another important point is that both projects shown here today focus a lot on profiling neurons, but we've also done a lot of work to profile other cell type populations within the brain, such as microglia and astrocytes. So please keep an eye on the Spatial Organ Atlas as we continue to release additional stories. We should see some human brain data coming out very, the data is there, the story is coming soon, and we'll see some additional segmentation type of approaches there. Additionally, I want to point out, if you have a GeoMx DSP and you're interested in contributing to our Spatial Organ Atlas data repository, please visit our website for more information. It's a growing resource, and we expect it will be a tremendous resource for the spatial biology community. With that, I would like to acknowledge the whole team that worked on the Spatial Organ Atlas data release at NanoString, specifically for mouse brain, along with our fantastic collaborators, Ellen Hess and Caitlin Romano at the Emory University School of Medicine. Thank you so much for your time today.
Well, thank you, Alison, for your informative presentation. We will now start the Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just click on the Ask a Question box located on the far left of your screen. So let's get started. So our first question that came in for you, Alison, is, can GeoMx Digital Spatial Profiling be used to spatially profile protein?
Yeah, great question. Absolutely. We offer neuroscience-specific modules for both protein modules for both mouse and human.
Excellent. Another question that came in is, can I study other species such as rat with GeoMx DSP?
Yeah, absolutely. So NanoString offers a custom probe design and barcoding service for up to 200 targets. And with that, you can design your panel specific to your experimental question for a rat or other non-mouse, non-human species.
Great. Another question that came in is, I see that you have a human brain data set in the Spatial Organ Atlas. When will you be publishing a Minerva story on this data set?
Yeah, absolutely. The data right now for our human brain samples is available for download, but the story will be available hopefully by the end of this month. So stay tuned and definitely check that story out. I'm excited about that one too.
Okay. Another question that came in is from Carmen with Royal College of Surgeons in Ireland. Is there a difference in RNA quality when using FFPE versus fixed frozen? Can you test RNA quality prior to probe collection?
Yeah, so GeoMx works with FFPE tissue, fixed frozen. The striatum samples from the mouse brain that we profiled were fresh frozen. So we have protocols for all three tissue preparation types. We do not see big differences in RNA quality, well, in signal quality from section to section with our GeoMx whole transcriptome readout, sample type to sample type with that readout. And then if you want to test the quality of your samples prior to probe collection, our assay is an in situ hybridization-based assay. So we really recommend that you use like a lower plex-ish assay to test the quality in such as RNAscope, look at medium and low expressors. And if you can detect medium and low expressors in your tissue of interest, you should be good to go with using the GeoMx RNA assays.
Great, thank you. Another question that came in is from Gabriel at LCSB. Did you sort the cells for IBA1? If yes, are you missing some cells that are in the amygdala but don't necessarily express the marker?
Yeah, so in this specific experiment, we did not collect from the IBA1 positive cells. So we looked specifically at neurons and then the neural pill components. We have done experiments where we've looked at microglia and have used a combination of IBA1 and TMEM119 for that type of segmentation. And actually, there is some microglia-specific segmentation coming in the human story.
Great. A couple of more questions from Gabriella. Did you take into consideration patch and matrix in the interior-posterior differences?
Yeah, so I'm not sure. I don't think I ended up including a full image of one of those sections that we profiled from the striatum, but essentially the same coronal plane was used for every single mouse. So we matched the anterior-posterior position from mouse to mouse to mouse that were profiled there. And again, really focused on the dorsolateral, dorsal-medial, and ventrolateral components of the striatum.
Great. Another question from Gabriella. How does this data compare to humans since the structure of the striatum differs between mice and humans?
That is a fantastic question. So I would really love to see some GeoMx data from human striatum to answer that question. Right now, I don't have a great answer. I think we would really like to see the comparable data from human, make a call from that.
Great. And another question that came in from Mark, you mentioned having a human brain transcriptome atlas as well. Did you or others see any significant differences, as have been recently shown in DRG sensory ganglia by Ted Price and others?
Yeah, so for our human brain transcriptome atlas, we profiled areas of the cortex and hippocampus specifically. So we don't have as many regions profiled within that data set, but we do go a little bit deeper into the differential expression between the different cell types in the different given areas.
All right, and another question from Carmen. When drawing contours, is it possible to do this in the GeoMx software, or does the file need to be exported into ImageJ? What file type, OME, TIFF, or is TIFF sufficient?
Yeah, so we have a contouring type script for ImageJ that can be used with our software to draw the contours. So the file type that you'll export, there's actually a specific export button within the user interface of the software specifically for exporting the ROI specific TIFF files. So you don't need the OME TIFF for that. You export individual TIFF files, run the script, import them directly back into the software, and then you can actually visualize the contours directly on your sample before you collect. So if you wanted to go back and do any additional editing in ImageJ, you could do that. You'll see exactly what will be collected.
Excellent. And then we have another question from Gabriella that just came in. What is the collection protocol for fresh frozen samples? How do you dissociate the tissue? My concern is always that I miss some cells that are more sensitive to damage.
Yeah, so I mean, one of the nice things about the GeoMx workflow is that it's non-destructive to your tissue. So we do fix the tissue. We do fix fresh frozen tissue actually overnight before we proceed with our sample preparation, which is pretty standard in situ hybridization preparation, and thus leaves the tissue really nicely intact. You can adjust your proteinase concentration kind of depending on your concerns for your specific sample type, but in our experience, that initial fixation is robust and allows moving forward with the processing of both sample types.
Excellent. Well, thank you so much, Alison. So does anyone else have any more questions that they didn't get a chance to answer? Now is the time. All right. Well, that concludes our Q&A session for today's webinar. Do you have any more final comments for our audience, Alison?
I don't think so. Thank you all so much for your time and attention. It's really exciting to get to share some of this neuro-specific GeoMx data with the greater community.
Thank you. So thanks everyone for your time today, and thanks Alison for presenting this research. Before we go, I'd like to thank the audience for joining us today and for their engaging questions. Questions that we didn't have time for today, and those submitted during the on-demand period will be addressed by the speaker via the contact information you provided at the time of registration. And this webcast will be available on demand for 2 years until March of 2024. Labyrinth will alert you via email when it's available for replay, and we encourage you to share that email with your colleagues who may have missed today's live event. And that is it, and that concludes the NanoString webinar for Labyrinth's Neuroscience 2022. Thanks everyone, and we'll see you next time.
Thank you.